Okay. Hello, everyone. Welcome to the third colloquium of this summer session. My name is Gyan Bhanod. I'm a faculty member at Rutgers in the Physics and Biology department. And I'm going to introduce our speaker, Dr. Alan Perelson from Los Alamos. Alan is famous for his work in virology. And if this were a virology conference, I would not be able to introduce him because everybody would know him personally. But he is one of the 6 non-retired senior fellows at Los Alamos, which is the highest honor that the lab can bestow to a scientist. Alan did his PhD from UC Berkeley, and he has been honored by many distinctions. He's a member of the American Academy of Arts and Sciences, fellow of the American Association for the Advancement of Science, the Society of Industrial and Applied Mathematics, the American Physical Society, and the Society for Mathematical Biology. So he basically covers many fields all at once. He's also the recipient of what is the equivalent of in physics, in biophysics, of the Nobel Prize. He got the Delbruck Prize in biological physics for his profound contributions to theoretical immunology, which bring insight and save lives. His research focuses on developing models of the immune system and infectious diseases, such as HIV, hepatitis B and C, flu, the Zika virus, and most recently, of course, SARS-CoV-2. Please, welcome.
What was this business area?
What was the thesis area? Biophysics.
Non-equilibrium thermodynamics. Yes. Thanks. So what I want to do today is give you an overview, first of a little bit of biology for SARS-CoV-2, and then discuss a set of models that we've been developing, which are all based on data. So what I'll show you first, usually, or concomitantly with models or data sets that we're using to inspire models and to try and validate models. After about the first 2 or 3 very simple models, it'll all be work that we're currently doing, so it's ongoing, just to give you a flavor of where we think this field is going. So let me start with a simple.
You have to say God is.
Okay, how do I get rid of that?
You click on God.
Click on.
Can you see it on your screen?
No, I don't see it on my screen.
So there's a.
Under there.
Okay. What if you minimize that? You'll see.
Enter.
Yeah.
Just hit enter. Okay, let's try that. Now it just goes back.
Try.
Right.
What if you stop sharing the screen for me?
It stops sharing the screen. We don't see this.
Get out of full screen.
Wait a second. Oh, here we go. Oh, I thought I just clicked on that.
It's behind this. So maybe you move it.
Sorry, I'm not a great person on Zoom.
Go out of presenter mode and whatever it is, the presentation mode.
Get out of presentation mode. Thank you. Where am I? All right, let's get out of presentation mode.
Let's go. Yeah.
Escape.
There you go. No, minimize this. Minimize that.
All right, let's minimize this guy.
It's behind Zoom.
No, I still haven't got. We have any experts on Zoom here? So I can see it here.
Where's the presentation?
Presentations, PowerPoint down in there.
No, it's still there. Choose that thing instead of picking on. Yes, choose that thing and maximize it, right?
All right. Maybe let me just close PowerPoint and start over again.
No.
Yeah,
That I can do. Let me start.
Escape from the presentation and get out of full screen mode.
No, that's not it.
There was a cursor there.
Oh, you seeing my cursor? I don't see it.
It's you move to the second screen right now.
All right.
Get out of full screen.
But you need to exit the presentation.
All right, so hold on, let me just get it. It's not being very nice. Ah, here we go. Got it. All right. Let's get that one. Let's start over again. Thank you, guys. Okay. Now we go into full screen presentation. Ah, come on. There we go. Okay. Well, I apologize. So anyway, as all of you probably know, that SARS coronavirus is what's called a beta coronavirus. It's related to a number of viruses that cause colds. It has a genome that's a single-stranded RNA, which is that strand in the center of the screen. And most prominently, it has spike proteins that are circled here. There are about 100 of them on the surface of this virion, which has a diameter of about 100 nanometers. This spike protein interacts with receptors on cell surfaces to infect those cells. And here's just a cartoon of that. So here is a cartoon picture of the virus with the spikes sticking out. Here's an ACE2 receptor on a cell, like an epithelial cell in the nasal passages of the lungs. That binding causes the virus to be attached to the cell membrane. And then there is a co-receptor, something called TMPRSS2, which has enzymatic activity, which cleaves the trimer, one domain of it called S1, and it exposes what's called a fusion peptide in the S2 domain, sort of depicted right here, which allows the membrane of the virus to fuse to the membrane of the cell. After that fusion event, the RNA that's packaged inside the virus enters the cytoplasm of the cell. It's a positive strand RNA, which means it's equivalent of messenger RNA. So ribosomes can get on it and start translating proteins and generate either full-length transcripts and also partial transcripts, which is where you get the various viral proteins being generated. When concentrations of proteins and RNAs get high enough, the virus starts assembling intracellularly and then is secreted from infected cells as virions. Now, the period of time from when the first interaction occurs until viruses start secreted, there's something called the Eclipse Phase of infection, and I'll just mention that in some models. What's very interesting about this virus is it makes enormous quantities of viruses from newly infected cells. This is an electron micrograph taken from the New England Journal of Medicine, where experimentalists got human airway epithelial cells, which have cilia on it. These cilia beat and move mucus up out of your throat. And all of these tiny little particles, maybe here seen in the expanded view, I don't know why I don't see my mouse very well, maybe you do, but all of these little spheres here on the right-hand side are virus particles. It's only one part of a cell. Here's a scanning electron micrograph of part of another cell, again, showing enormous quantities of the virus on the surface of this cell. And I'm guessing there's tens of thousands of them have to be secreted by an infected cell.
Oh, we only need to bind with a single receptor, though, right? We don't need nothing cooperative.
Nothing cooperative, as far as we know. These viruses then can be shed off the cell surface and infect other cells. There was a report about 2 weeks ago that the virus may also be able to move intracellularly. So from one cell to another within a cell layer, like a tissue of the epithelium. So it's a preprint on medRxiv. So not reviewed yet and not validated. The infection occurs usually first through the upper respiratory tract, which is the nasal cavity, the pharynx, the larynx. And then viruses can be transported down into the lower respiratory tract through the trachea and the bronchi, and then finally into the lungs and the alveoli. Usually when it's in the upper respiratory tract, one has typical cold-like symptoms. You may be coughing, sneezing, not feeling well. Once it gets into the lower respiratory tract, the disease can become more severe. It can start killing alveoli and airway cells, and you'll have a more severe severe disease. The virus is thought to be shed from the upper respiratory tract when we speak, when we sing, when we cough, when we sneeze. And here's one of the famous pictures of somebody expelling the droplets. That's been in the literature for a long time. Adrian Box, who was supposed to be here, is an expert on that. And he's actually done photography, high-speed photography and imaging of events like this. But it's the upper respiratory tract in which the virus is being spread from.
So much deadlier than other coronavirus.
Why is it more deadly than other coronaviruses? The general thought is because it's novel and we don't have very much of a prior immune response. So the possibility has been raised that people who have less severe and mild disease may have some cross-reactive immune responses with cold viruses, but the people who have more severe disease may not. They also may have some sorts of immunodeficiency due to underlying conditions. And don't forget, about 80% of infections are either asymptomatic or very mild. There's only sort of over 20% of infection of people who get infected have severe infections. And of that, maybe it's only 5% that get hospitalized. And as we heard the other day, the death rate is really about 2% of cases. And there's all sorts of comorbidities that are involved with that.
As far as this particular process, so just the cold coronavirus would be expelled in similar size particles,
Yes.
in similar survivability and those kinds.
Correct. Correct. That's at least my impression. And the other viruses in this type actually were more deadly. MERS, if you remember that. So depending upon the exact way that these viruses work and infect cells and how much virus they produce per cell, how long the infected cells can survive during the infection, those things differ from one virus to another. What's interesting about this virus and most respiratory viruses is that they cause acute infections. So, typical period when somebody is, quote, infectious, and, as you know, from periods of isolation is something like 14 days. And then the infection is generally cleared, but not in everyone. And the question then becomes, why are some viruses give acute infection, some chronic? And this is really a general problem. And there's three main explanations that are given. One is called target cell limited. So we think that there's only a limited number of cells that this virus can infect. So I mentioned that the cells have to have the ACE2 receptor and co-receptor like this TMPRSS2. It's been estimated from RNA-seq experiments that the fraction of epithelial cells in the upper and lower respiratory tract that express both is something like 1%. There's been work by a guy named Ralph Barrick at the University of North Carolina who's done more detailed studies, and he thinks that has evidence that there's a gradient with cells up in the nasal passages maybe having 20% of the cells having these receptors, while as you go deeper into the lung, it can get to 1% or less. And of course, the lung has a lot more cells in it than your nose does. So that's what we call target cell limited. It's like an epidemic, you run out of cells to infect, and then the infection sort of dies out. The second thing that's involved is something called an innate immune response. So cells, or the immune system has both chemical responses early on and cellular responses that develop very, very early. They secrete chemical mediators. And one that we'll talk about is innate immune responses that can make cells refractory to infection. So it causes upregulation of genes that makes it very, very difficult for the virus to replicate in them. So it's a way really of expanding the target cell limitation. So cells that generate this innate response, if they're not able to be infected, that also limits the targets that can support replication. And lastly, starting usually about 2 weeks after symptom onset, you start seeing substantive adaptive immune responses. That means antibodies are being detected in the circulation, usually starting somewhere initially, maybe day 7 after symptoms. By day 14, you'd start seeing reasonable amounts, T cell responses start coming in. And one general view of acute infections as that target cell limitation and these innate responses handle the virus infection early on. Very few of these infections really can be cleared by that, and that later on the adaptive immune response comes in and cleans everything up. And there are various examples of other viruses that we've studied. So the simplest model of acute infection is one that my group introduced in 2006 when we were studying flu. And this is a prototypic target cell limited model. There are a set of quote targets, which are represented by T here. They become infected by virus particles, represented by V in the equations, and beta is an infection rate. So it's very similar to an SIR type model in epidemiology. We have omitted replenishment of the target cells because in these acute infections, the viral infection is so rapid that there's not very much time for regeneration of target cells. Normally, in the absence of infection, there's both production of target cells and their death, and they're generally in balance. And then this infection comes along and perturbs that. And so the assumption here is that the production and death in the simple model are in balance. We've looked at models where we add in terms, and I'll talk about that later on. The infected cells arise, they get killed by a combination of natural death and cytopathic effects of the virus. It means the virus kills the cell. And this parameter delta for the rate of death per cell in these cytopathic viruses is overwhelmingly determined by the rate at which the virus kills cells. And then lastly, in this simple model, we assume that each infected cell can produce virus at some rate P per infected cell, and that virus gets cleared by a first order process. And that's actually been measured in a number of infections, not in this one, but we assume that is the case. Now, this model fits data very, very well for SARS coronavirus. It's missing this Eclipse phase that I spoke about. It's the same in the case of influenza. You can fit data very well without it, but then you get estimates of the lifespan of the infected cells that don't really agree very well with data. So we think it's important to include this extra Eclipse phase. So cells I1 become infected, but they're not yet producing virus. And then it moves into the second stage, here represented by I2, where we say cells produce it. And this is just an exponential residence time. It's the simplest one. There's been enormous work in this field, putting in gamma functions, distributed delays, Erlang functions. You like it, and depending upon the resolution of the data, you might get some clues as to what that model is. But Eclipse phases are usually on the time scale of hours, and I'll show you experimental data. That's their sampling is nowhere near that frequent, and it's days and weeks. So it's in there, but it's not terribly important. What can compute from these models is something called the basic reproductive number, but this is intracellular. This is not the basic reproductive number we talk about in epidemiology, but it has the same interpretation. We're interested in how many secondary infections occur if you have one infected cell in an environment where all the neighboring cells are susceptible to infection. And you can compute it algebraically or exactly from these model equations. The numerator is basically everything positively causing virus expansion, and the bottom is sort of the clearance. I'll show you estimates of R0, and the objective of vaccinations, immunotherapy, therapeutics is to reduce some of these parameters so that the R0 value goes less than 1. It's like taking a branching process and making it subcritical, and the infection will die out. So that's fine. Those are nice toy models. They were inspired by influenza and some other viruses. So does that match SARS coronavirus too? So we've been looking for longitudinal data to compare with models. And this is one of the early papers from March 2020, in which a group of patients were followed longitudinally in Singapore. Here's a set of patients where there's a reasonable amount of data and they weren't under any sorts of treatment. And you can see the data really starts at the time of symptom onset or later. We'd like to start our models from the time of infection. And there's epidemiological data suggesting that what's called the incubation period, the time from infection to symptom onset, has a median of about 5 days. So this model just didn't have any early data, but we set time 0 to be 5 days before the patient reported symptoms. And then you can see the data here is all those black dots. The horizontal line is what's called the limit of quantification of the assays that are used to determine the virus. So below that dashed line, any points that are on that are just below limits of quantification. They're usually just put at that limit. It's not that the virus has gone away, but that's basically 100 RNA copies or 100 virions per ml of whatever has been swabbed. And you can see the model, excuse me. Are they all symptomatic at the same time? We don't know. At the same level. Well, you can see that in this experiment or this data set, The peaks tend to run from about 10 to the 4th copies to 10 to the 6th. There was a large study that was published in PNAS by one of my colleagues looking at something like 1,300 patients in France, and their distribution went up to 10 to the 10th copies. So if you study enough patients who have more severe disease, you can have much larger peaks. So from this study, which only had 13 subjects, that we fit it to the simple target cell model, we went with the Eclipse phase, they both fit. We use something called a mixed effects approach, where we simultaneously fit the data from all the patients and try and estimate parameter distributions.
Can you say again what Eclipse phase is?
That's the time, that's like being exposed, but not yet infectious in an SIR model. So it's the time between when the virus first interacts with the cell before that cell starts producing virus. So uncoding, translating the RNA, making proteins, assembling viruses. We estimated here that the R0 is about 8.6. So that's the number of new cells that one infected cell would infect on average. Lifespan of cells here, is of order of 1 day. So that's one of the reasons that this is become a serious viral infection if it's killing the cells that it infects within an order of a day or so. So then we asked at this time, early in the infection, there were a number of agents that were being hyped by our president and others. Hydroxychloroquine was one. Remdesivir was just being started. That's an RNA polymerase inhibitor that was developed to study to fight Ebola. And it was, and then the idea of having monoclonal antibodies or convalescent plasma was starting to take hold, and there were some studies taking plasma from people who had recovered, it's called convalescent plasma, and sticking them into other patients, hoping that the antibodies there would do something. And we can model that or include those effects by introducing the idea of an effectiveness. So we say, in fact, effectiveness of a drug or therapy goes between 0 and 1. 1 means it has no effect. 1 means it's 100% effective. And for example, if we have a drug like a neutralizing antibody or hydroxychloroquine, which was thought to block infection, we just take the infection rate data and we multiply it by 1 minus epsilon. So if the drug's 100% effective, the infection rate goes to 0. And the same thing for drugs that will block viral replication, like a RNA polymerase inhibitor, which is what remdesivir is, same thing. And then you can also envision increasing the rate of viral clearance or increasing the infected cell death rate. And I don't have time to go through explain the immunology behind that, but if anyone's interested, we can talk about that later. But they're increasing, so we divide by 1 minus epsilon. We found early data on how effective these drugs might be by assuming that the effectiveness was related to the concentration in a saturating way. And that's very common in pharmacodynamics. And you can use a Hill function or just a simple Michaelis-Menten-like phenomena, where in the denominator you have a concentration of the drug divided by the concentration plus its effective concentration 50. Looking at data from the literature to understand how the concentrations change, we calculated what the average would be over 7 days, which would be a typical time of giving these therapies. And you can see, for example, that hydroxychloroquine, we calculated would have a 6% effectiveness, so barely any effect at all. And in fact, the only drug of this list that looked like it had some promise might be remdesivir. And these estimates were all made in vitro, and we felt that the drug concentration was likely to be lower in vivo because the drugs can bind to various proteins, not all of its bioavailability available. It may not be able to penetrate tissues. So we weren't that excited about remdesivir either. But anyway, that's the only one we thought might have a noticeable effect. And we also, let me just finish this one sentence. I don't want to show you this, but by simulation, it was very easy to show that the most benefit would be achieved if the drug is given as soon as possible after infection, preferably before the viral load peak. And that's because once you hit the viral load peak, the virus is coming down exponentially already, and adding a drug to reduce the virus further doesn't have very much effect, because also near the viral load peak, most of the target cells are already infected, which is why the viral load is dropping precipitously from that. So the other option was to use or think about giving these drugs prophylactically. So that means somebody in your household is infected, and then you start taking it, or you're At that time, you're an ambulance driver or a public health safety person, or you're in a nursing home and somebody's infected. If we had drugs, you might start giving them prophylactically. And I should say, this is on my conclusion slide. According to the New York Times today, Anthony Fauci has committed, the government has committed $3.2 billion to develop some antivirals that you can take as pills, so someone can actually take them very early on. Whether they'll be developed and distributed, we don't know. We've been involved in a collaboration with some clinicians associated with Harvard Medical School, and had access to data from hospitalized individuals where viral loads were measured in blood and in various swabs. And what we concluded from that, some of the patients were on remdesivir and some weren't. And as you can see here in that bottom picture, in the patients that were treated with remdesivir, which is up here, the estimated viral decay rate was about twice that at which the virus decayed with untreated patients, which was given in blue. So it looked like it may have some activity. The sample sizes are small. And the other interesting feature, which came out in discussions here earlier in this week, is this is one of the few studies that actually looked for virus in blood. And by the time symptoms had been onset, there was virus detected systemically in the blood.
I'm wondering for the people who get seriously ill,
Yes.
do they still have this exponential decay that their late symptoms are later on, or does the viral load stay?
I don't know, to be honest. I'll show, because most of the data that we have for patients that are not severely ill, patients who are severely ill, they're trying to keep them alive and they're not being sampled every day. And it's also, they're quite infectious, so it's hard to get that data. But I'll show you that at the end, I'll show you dynamics that go on in the lower respiratory tract and lungs, and it's sustained much longer. And it's somewhat interesting. If you guys let me get there. Yes.
I have a quick question. So I also have a colleague that does these types of models and stuff. And I think one of the biggest problems he brings up is, are we getting the right numbers from the clinicians to be able to train some of these models in some cases? And is there something we should be telling the clinicians that they should be reporting more?
Right. So again, it depends on the data sets that you're looking at. I usually have more confidence in data sets like the first one I showed you, where all the data points sort of line up fairly well with the models. If the data points have a huge amount of noise and scatter, then I have less confidence in how they're being collected. But that's just a personal bias based on prior experience. So in terms of trying to do prophylaxis, or interested in starting therapy very, very early, I mean, deterministic models are not really appropriate. There, what we're interested in is you encountering a few infectious virions, and various clinicians have estimated, or guesstimated, I should say, that it might take somewhere between 1 and 10 infectious viruses to get this thing started. So small numbers. So it seemed much more appropriate if one wants to study the very early stages of infection to come up with a stochastic model. So this is just a translation of that ODE model into a stochastic model. Because we're interested in early events, we have set the number of target cells T just to the initial number, T0, and that linearizes it. We've also done simulations where you allow that to be nonlinear, but the approximation is extremely good. You can write those equations then in terms of a master equation or branching process. They can be solved analytically when T is constant. If the extinction of the infection is just a multidimensional analog of the classic Gamber's rune. We've published, some colleagues have also published solutions of that model, which references are given down there. And you can see here, phi here is what we call the establishment probability, 1 minus the probability of extinction. R0 here has been changed a little bit because now we account for every single virus particle. So we also consider the possibility that the virus is being cleared by infecting a cell. Generally, we leave that out because one virus goes into a cell, and then if it makes 10,000 other particles, it's not really worth your bother to deal with that. But in very early stage, with a very few particles, that might make a difference. So the R0 has an extra beta T0 term. And then, as you might expect, if R0 is less than 1, then the establishment probability is 0, or the virus will go extinct. And if R0 is greater than 1, then there is still a probability that it'll go extinct or go established. And the larger R0 is, the higher the probability that it will become established. And also the larger the inoculum dose, more infectious viruses, the higher the probability. If R0 is greater than 1, that it'll continue. And that probability depends on R0, the inoculum dose, and also parameter N, which is we call the burst size. In the models, it's the production rate per unit time divided by the multiplied by the average lifetime 1 over delta, which tells you the number of virus particles produced by the cell over its lifetime. So those are the only parameters that become important once you know R0. Then you can do what I had spoken about earlier. You can take these parameters, beta for drugs that block infection, or P that block production, multiply them by 1 minus epsilon in these functions, because the beta occurs both in the numerator and denominator. It's a little fancier, and you have to look at this function. But anyway, you get an analytic expression for what's going to happen for the establishment probability in the presence of drugs of epsilon. And the most interesting finding is that there's something called a critical efficacy, which really corresponds to driving R0 below 1. And you can see that in these graphs where the establishment probability, when the drug efficacy gets to be something like 0.9 or close to it, all of these establishment probabilities go to 0. That means you're above the critical efficacy. Before that, the probabilities depend on initial viral load, which are the 3 sets of curves. And you'll also see that drugs that block infection, which are in yellow, are more effective at reducing the establishment probability. And that's because if you prevent a cell from becoming infected, that's sort of the best you can do. And it's much better than just reducing the amount of virus it produces once it's infected. There are other things you can do, I just don't have time to go through, but you can look at them in this paper, because you also sort of flatten the curve if you look at the real dynamics coming out. So as you increase the efficacy of the drugs, even if they don't prevent infection, they drive the peak down, they flatten it, and they allow more time for the immune system to take effect. They also cause problems, because if you're infected, it may take weeks before you actually develop symptoms if you're on these drugs, because they're slowing down the infection so much. So there's some issues related to that also, which we raise. Anyway, now let me show you another set of data, which is one we had been drooling over or wanting for a long time. All of the clinical data published up to this paper was always using patients that had symptoms and sort of got hospitalized or went to doctors and somehow got swabbed. As you may remember, the National Basketball Association last year took all of their players, brought them to Miami, and put them in something that they call the NBA bubble. So they were all isolated. And in addition to that, they were being swabbed roughly daily or every few days to make sure that they were testing negative so that they could play basketball. And of course, some of them did get infected and tested positive. There's a data set that's in this preprint, just like all Jonathan Grad from Columbia is a senior author, and my colleague David Ho had gotten the NBA to do this. He's also another author. In this preprint, they have measurements from 68 individuals where there's more than one pre-peak viral load, and we're doing some work on that. But here I'll just show you the data from 9 newly infected individuals where we actually can see the upslope, and I'll show you that in a second. We fit these target cell limited models to the data, and then we also threw in a model with an innate immune response because we thought the data could support that. So the data with the innate immune response, which is something I spoke about in the introduction, is we say that infected cells can produce innate immune mediators, something like interferon, that can lead to cells becoming refractory to infection. So that's our refractory population. So target cells become refractory. As the viral load and infected cell levels go down, they can return back to being target cells. So there's just that one extra bit. And what we found is that that model proved better than fitting models that are strict target cells. And here's fits to those 9 individuals from the NBA bubble. There's log likelihoods for the 2 models. The dashed lines, which are hardly visible except in the middle one, 1740, and the one to the right, the dashed lines of the innate immune response model. And they prolong the infection because they're putting cells into this refractory state and then they can come out again. We don't have any data out there, but even without that, they fit the data a little bit better.
Can you go back to one slide? Model?
What?
The model is only beta, except for those stochastic parts, it's only beta T that comes in here. So you don't get any beta and T separately?
No. Well, T comes in, for example, in the second term in T, but beta always comes with T because you have to infect cells.
Yeah, and so what is the ballpark of how big T is? What is that?
What's the ballpark of beta?
How many target cells?
So from these models, that's not identifiable. In fact, what you can prove from the basic target cell model, I think it's true here also, that the product of the viral production rate P times T0 is the only thing that's identifiable. And you can do that by dividing through by T0. All the things that have to do with cell populations, that doesn't have much effect until you get down to the viral equation. When you divide I by T0, then you also have to multiply by T0 and you get a pi times T0. So we know we can't estimate that. And in most of these models, if you change the target cell level by a factor of 10, then you get a increase or decrease in the production rate. You have more cells, ultimately they'll be infected. You can also do something like that with beta and T and shows that also may not be identifiable in an easy way. So the guesses for target cells really come out of the anatomy. So there's ancient literature about how many epithelial cells there are in the upper respiratory tract, number of what type 1 and type 2 alveolar cells in the lungs. We use those as starting points, and that gives us an estimate of these production rates. But what I didn't say, the reason we changed from P to pi here is we're starting to look at this data. We also recognize that you're not really being able to sample all the virus. You're always just taking a fraction of it on a swab. And it turns out if you take that into consideration, what you find is that, take that into consideration, going from number to number on a swab or number per ml, then you get the fraction of viruses that you're actually sampling times the production rate, which is what we're calling pi. These are all technical details which give us grief, but let me get on to go over the major points. Claire? Refractory means the cells aren't subject to infection.
Okay.
So they're in a quote an antiviral state is what immunologists say. So think of them as cells that possibly have gotten stimulated by interferon. It turns on 100 or more interferon stimulated genes. Some of them turn off something called PKR will turn off translation. So the cell will not translate any proteins, or essentially not, until the viral levels and the interferon levels go down. So anyway, the virus is fit. So but that was only 9 patients. And now, and we tried to estimate the day of infection. They didn't know that. If you looked at this data, it's all normalized. Day 0 here is just the time of the peak because they're sampling. And before that, they're just getting negative, negative, negative. And these are the first positives. And same thing at the end. Here's limited detection. But if I showed you the full time series, they may be continued to sample for many weeks. And there's just no detectable data. So what was interesting about this paper, which is also an early one from March 2020 by a group in Germany, headed by Christian Drosten, who's sort of the Anthony Fauci of German medicine, is that they assessed the earliest cases in Berlin, where somebody from China came for a business meeting, infected somebody in an office, went back. A few days later, this person got infected. This person had interactions with other people in the office. They started getting infected. And the Germans, out of great caution, took everybody and hospitalized them and sampled them like crazy. And they also had some idea of contacts of who contacted whom. So here's 2 data sets. There were 8 people in that German data set with reasonable amounts of data, which is shown on the top. It's all post-symptom onset. And we're just estimating the time of infection. 0 here is being estimated by model fits. And we're simultaneously fitting that data along with the NBA data. Oh, excuse me, the German data, we know the time, the epidemiological data from contract tracing, they actually publish when they think the people were infected. So we think those are the real times. The other things that's interesting for those of you who can see it, there's a vertical line in all of those plots. That's usually before day 5. That's the time these people said that they had symptoms. So it was very, very early. And and then they were isolated, so they didn't really spread things. We were trying to calculate things like serial intervals from them, but it's not very realistic because they were very quickly isolated and didn't interact with anybody. And then the bottom is the NBA data, and for reasons I don't really understand, they never asked people when they started feeling symptoms. It's just, they just never asked that question. So they did say something about at some point they were symptomatic, but we don't know when symptoms occurred, and their data is very, very scarce. And we're analyzing at least based on whether they were symptomatic, but I don't have that analysis isn't totally finished yet. So fitting the model to all of these things, we get things that are very similar to our first initial target cell limited model. The average Eclipse phase length was about 6 hours here. The death rate of productively infected cells gave rise to an average lifespan of about 14 hours. So the total summing those is about 20 hours. So again, I said, these cells are living about a day. The first 6 hours, they weren't making virus on average. And the R0 here was 7.4 versus 8.6, and nothing to write home about. So we think that these cells, infected cells, can infect neighboring cells with a wide range, but something in the order of 7 or 8 on average. Now, the next thing that interested us and that interests everybody is, can we relate the amount of virus in the upper respiratory tract to the probability that a person will transmit the virus and infect someone else? So we came up with a toy model of virus transmission. So what we're going to say is the donor, somebody's infected. He has a combination of infectious and non-infectious virus, and that he has a contact with somebody in a supermarket parking lot, his household, and we'll consider short contacts of length of minutes to hours on a time scale such that the viral load in the donor we can take to be constant, at least at this point. So it's not critical how long the contact is, as long as we just keep the viral load constant. And then we say that of all the viruses in this upper respiratory tract, a fraction of those are going to be shed per unit time. And that's going to be mu times VI if you want to read all this. VI is the number of infectious viruses, because infection, what's important is the number of infectious viruses that actually make it over to the recipient. And then we're going to say, no matter how many he sheds, which we will calculate here, that there's going to be a fraction of them that are infectious, and a fraction, excuse me, here's just all, take that back. We're just looking at the infectious virus for a moment. We'll say a fraction of the ones that are shed get to the recipient, fraction phi. So the number is in yellow there, you can calculate it. It's basically a constant times VI. That's an average. We assume the actual number that reaches any person is Poissonly distributed with that as a parameter. And that of those, there's a probability nu that a virus that reaches the recipient will actually trigger a successful infection. So we take a binomial distribution over those, and then since X is Poisson, you can calculate that the whole thing becomes Poisson, and again, with a parameter that's going to be this thing alpha, which is all of those constants. So the important thing is you end up with a Poisson with a constant times the number of infectious viruses in the exponential. So a trivial model, people experimentally have seen things of that sort. So then it behooves the question, and somebody should I raised before, how many viruses are infectious? What we measure in the individual is the total amount of virus, total amount of viral RNA. And virologists have generally assumed and reported that's just the constant fraction of V. So if you read some of Rob Phillips's recent PNAS paper with Ron Milo about various numbers, and or some of the other literature, what people do is they take an amount of virus in the lab, they measure the amount of RNA, and then they take some of that virus and put it into cell culture and ask what fraction of it can infect cells in culture and how many cells. There's various ways of doing that, things called plaque assays or TCID 50 assays, and I won't do that. But what they find is that in general, that there's some fraction, it's been reported to be between 10 to the minus 3 and 10 to the minus 4. There's some other data that I'm going to show you in the next 2 slides that suggests that it may be sublinear. And we tested both the power law and a saturating function. And let me just show you the data. So these are 3 experiments done in a slightly different way. This first one on the left is a heroic one. This is a group by Jaffa and all. They obtained about 4,000 swabs from patients with different viral loads in different stages. And they measured the RNA in all of those swabs. And then they took the equivalent amount of virus per ml and put it into cell culture dishes. And some of them were replicates. And what they then observed is the fraction of their cell culture dishes that they put that viral load in that became positive, that they could detect that there was viral infection in. And so cell culture positivity is the fraction of all the cultures done with that particular viral load that became positive. And Kurt looks very confused. And
This distinction between infectious and non-infectious viruses, these are all things that superficially look like
They-
SARS-CoV-2.
oh, they all look this, well,
I
we
mean,
think
They all
so.
have the same RNA inside them, they have the same proteins, but somehow some of them are
Yes,
isolated,
yes.
live and the
Yes,
others aren't.
so that's true. So that's true of all RNA viruses. I don't think it's been examined in great detail, or at least I haven't seen any examination for this. But so if you look at HIV, my favorite virus, and you look at the viral sequences of the RNA inside virus particles, you find the vast majority of them have deletion mutations. Some of them just fragments so that they can't code for various enzymes and proteins. Some of them have a loss or a mutation, something called a packaging signal, which is what's needed to actually get the RNA to interact with the other proteins to get packaged into virus particles. So there's all sorts of ways that viruses become non-infectious. It could be that the amount of protein that actually gets expressed in cells when they're infected is not sufficient to build all the spikes. So lots and lots of reasons that we can go into, but looking at them, you generally can't see them, see differences.
Bottom line for the fraction of the stuff that you spit out that's infectious.
Right.
You stated the number.
Oh, so numbers that you see when they do just a single viral load and a single assay, it's usually something like a few times 10 to the minus 5 to, excuse me. Yeah, something between 10 to the minus 3 and 10 to the minus 4. Okay, and then also in clinical samples, some of the clinicians will report, patients that have, I guess this Willful paper says that patients who had less than 2.5 times 10 to the 5th viruses per ml in a swab, when they tried culturing that virus, just from a couple of patients, they couldn't get any cultures to be positive. So that's where they get these numbers from, 1 in 250,000, 1 in 10,000.
Following up, what is exactly viral load? How is it exactly being measured?
What is the viral load?
The viral load? How is it being measured?
Oh, it's measured by PCR.
Or viral RNA.
Viral RNA by PCR, just as they do in all the other data that I show you. So it's taken from swabs. Swabs are put into medium. The RNA is extracted, etc. And for the culture infectivity, they don't extract the RNA and all. They just put the viruses in. So the simple linear model, the one that says it's proportional, fits the data terribly. If you look at the Jaffa data, it's in the dotted line in my cohesions. I hope you can see that. It's sort of an S-shaped curve at the bottom, and same in the other two models. It doesn't fit at all. The saturation model, which means a Hill function, and the power law, which are the red and blue solid curves, fit reasonably well. For the Jaffa data, the saturation model fits better because if you look at the viral loads between about 10 to the 7th and 10 to the 9th, where there's lots and lots of samples, all of them line up at exactly the same place at a little bit over 0.8. And then there's only a few samples that were actually measured that had viral loads of 10 to the 9th and 10th, which is why you have these huge error bars. They're very rare to find. The power law will actually go all the way up to that. And the second data set, as you can see, the power law and saturation sort of agree up to something like 10 to the 8th viral loads, and also a new data set that just recently came out, James Jones et al. So we forgot about, eliminated the linear model, and we've done analysis, both the saturation function and the power law, and they don't really make terribly much difference from what we've done so far. So I had that little transmission model where I had e to the minus a constant times the infectious. So now we put in these power laws or saturating functions, and if you do it right, there's other constants. So you get new sets of constants. But the whole point is it's just a constant either to a power law, a constant to a Hill function. And when we fit all three sets of data with the power law and the Hill functions, the constant H comes out between about 0.4 and 0.6 For our best fits for the Jaffa data, it was 0.51, and for one of the other ones where we had the power law, it was 0.45. But it seems somewhere in there between, at least these three sets of data, there'll be somewhere between 0.4 and 0.6. For the saturation model, this parameter theta tells you sort of the maximum probability of transmission. So if V goes to infinity here, the probability of transmission is 1 minus e to the minus theta. Turns out theta we think is small. That means it's approximately theta. So theta is approximately the maximum probability of transmission. And then we just looked at some epidemiological studies that tend to put that number at something like less than 20%. So we just, the slides I'll show you now, we just fixed that at 20%.
If two people, one of whom is infectious, the other one susceptible, if they get an opportunity long time, this is a 20% transmission rate.
You're all mumbled because you're mathematician.
Oh, sure.
Sorry.
If I want to know what the state is. So if two people were together for a very long time,
Not very long.
saturate to 20%.
Not very long. So,
But it's saturated to
in
20%.
the epidemiological studies.
Yeah.
Yeah, so this could be from secondary attack rates in family settings, for example. I'm not a great
Yeah,
epidemiologist.
but I mean, but as far as the model you have, these
Yeah.
very parameters come in, one of them is how long they're together for.
Yeah, but that all gets
It will saturate.
right, but in our little toy model, that all gets into a constant with all these other things. So
right.
the fraction shed and the fraction that gets transmitted, so we can't isolate any of those, which is one of the advantages for the theory. Just a constant times
Why would one expect it to saturate something as low as 20%?
oh, for real transmissions?
Yeah.
Well, that may very well be have to do with the immune systems of the recipient, how long the contacts are. But that's not everybody gets infected for a variety of reasons that are still trying to be worked out. And it may also have to do with the typical lengths of contact. So I'll mention 5 minutes. So one of the unique features is we get these probabilities of transmission for contact. But then since we know the viral load profiles for these 17 individuals, this is the more interesting one, is we can compute how the probability of infection under infecting someone else is a function of time post-infection rather than just viral load, so all the assays are. And you can see that these curves tend to be fairly wide. So there's a fairly long interval where there's a chance of you infecting people that you encounter. Which is, I think, somewhat novel. And the differences between the two models are pretty negligible. The other thing you can look at is the German data set. We know when they started showing symptoms. So you can draw a vertical line on these curves also, looking at the pre-symptomatic and post-symptomatic part, and then calculate the area under the curve where they were pre-symptomatic, and divide that by the total area under the curve. And that gives you the fraction of the transmissions that were pre-symptomatic. And at least in these individuals, we found that varied between 0 and 20%, and that it correlated with the incubation period that we were able to identify for these individuals from the time they were infected to they were symptomatic, which clearly makes sense. It's trivial here because you're just integrating over a longer period of time. But there are epidemiological studies that have also reported exactly the same thing, that people that have longer incubation periods have more asymptomatic transmission. The other things we've done to validate this is you compute from the number of contacts per day, which has been reported from European studies. This amazed me, was 13.4 from a number of studies. I can give somebody a reference. So we said that the R0 for an epidemiological study is 13.4 times the integrated probability of infectiousness to give you an estimate of R0. And that we averaged that over our 17 profiles, and that came out to be 4.3, which is in the range predicted by epidemiological data. We can predict the serial interval, which is just doing that integral now over time, with times time, to get the average time until you're infecting somebody. And that turned out, again, over these 17 individuals to be 6.4 days. And that's also consistent with estimates where patients aren't isolated, and which came out to about 6.6 to 8 days in some of the early data. This is the last thing I wanted to show you in a couple of minutes. Now, as I said, the German data also had lower tract data. It's interesting to look at it. And so what we did is we took our upper respiratory tract models, basically reflected them into lower respiratory tract and coupled them by transport of virus from the upper and lower. And this is the target cell limited model. We did that with the innate immune response model, which actually fits data a little better than this. And then we also added two other wrinkles, which I'll show you from the data in a second. So this is our best fit model. But first, just look at the data. The lines make it. So blue is the lower respiratory tract. So the red is what you've already seen. First observation I found striking, if you look at the early time points, they were detecting virus both in the upper and lower respiratory tract, roughly simultaneously at the first time these people were sampled. So it got into lower respiratory tract. Secondly, the lower respiratory tract infection in blue persists quite a bit longer than the upper respiratory tract. So all of these nice things like in the NBA bubble, you see it just decaying away exponentially, doesn't mean the virus is gone. At least that's the inference from this study. As you can all see, like if you look at number D, it looks like one of the bubble patients. We had the rest of the data, except it's persisting for another week in the lower respiratory tract. The third thing that really is striking, I think, is in many of the patients, you have a secondary peak coming up in the data. So if you look at A, you can see it goes down by day 15, then it comes back up. The same thing a little bit in B. At F is another one. In G, there's only one data point, but our model goes through it. And what we think that may be indicating is that the virus may find additional. So we hypothesize, and this works in these models, that it finds additional sources of target cells. So we can get that either if we put in continuous supply of target cells as source in the T equation, which I had neglected to get the acute infection, or if we just say at some point, which we then try and fit, that the virus moves to a new location in the lungs where it finds new target cells. So it's more of after some period of time, there's a delta function source, so it's now found that, and both of those fit that model very well. The delta function slightly better, but it's not enough to decide it. So we think it may have to do with the spatial area. And the third thing that we did here is at the very end, where there's no data and you see the viral loads going down to 0 in those dashed lines, is we invoked an adaptive immune response. So if you're out by day 21 or so, you know there's antibodies there. There were some antibodies that were isolated at those time points, although not quantitatively. So we just put in an increase in the death rate of infected cells starting at some point. So a fudge, but it's roughly what happens. So let me stop there. What I tried to show you is that models give you insights into the viral infection dynamics and possible effects of the innate response. Models with more detailed adaptive immune responses are under development, as well as models of later stages of the disease that involve the inflammatory response. So the virus can clear, and then there's sort of a second stage. It's all driven by cytokines and inflammatory responses. It's not virus, but other things going on that actually can lead to the death of the patients. Modeling of this type is very data limited, especially for humans. So there are animal studies also being done in parallel where you can biopsy them. And, they don't have anything to say about being sampled. But again, there is no perfect animal model. The closest that actually has symptoms is a hamster model. Non-human primates, you get good viral loads, but they don't get sick. So it depends on what you're looking for. And we have data from ferrets, which are the model of choice for ferrets. I can show somebody, and you also see very similar to the NBA bubble data. And they, for reasons not understood, they only get an upper respiratory tract infection. They can't induce infection in lower respiratory tract with human virus samples. And then studying the effects of therapy is important and something we're heavily involved in. And as I said, Fauci said we're going to try and develop pills. So let me stop. I'm over time, but we can talk now. We can talk over the line. But so let me take pressing questions. And
Yeah, we have only less than a couple of questions, and then we go for the one.
So for the last year, universities were taking samples of every student every 3 days or something like that, right? And then once the students get infected, they're re-sampled again with more specific PCR data. So can you, I suspect that universities would be very interested in sharing this type of data.
Oh, it'd be fantastic if they would. You know, there's all these privacy issues and data transfer, but if any of your clinicians would like to share it with us, we'd love to model it, or we'll share our models with you if you'd like to. get access to it.
We currently have not been able to, like, all the samples, the data that shown are like 5 people or 10 people, right? It's not 10,000.
Right. But even if you sample large numbers, not every, there's still only a small fraction that you're going to test positive and get enough samples. So it also depends on what their turnaround time is. So if someone tests positive, if they start sampling every couple of days, that would be great. But if they're sampling every week and then the results come back a week later, you may not get some. We're collaborating with somebody at St. Jude, where everybody in the hospital is getting, all workers get sampled once a week.
Certainly doing twice a week with a day.
Yeah. Yeah, I mean, I'm sure there are data sets. We just don't have availability. We've had the NDA just through David Ho and his collaborators.
Yeah.
Okay, you're gonna have to take your mask off and speak louder.
Does the severity of disease also relate to viral load mechanism as you just showed? And if yes, then does it mean that patients with lower severity or asymptomatic are less likely to get?
We don't think so. So if you look at the viral load curve, very preliminary stuff that we showed you, symptomatics and asymptomatics. The viral load curves are very, very similar. There may be some differences in the tail after the peak. So it's not at all clear that it's just the viral load that's determining whether you're symptomatic or not. Probably a lot of things having to do with host responses and innate responses in the tail. But we don't know. No one knows.
One more therapy. Are they looking for some drugs that target H2 or block H2?
You say blocking entry?
Yeah.
Blocking
Oh, H2. Oh, yeah, people are trying to block H2. There are agents that will compete.
Yeah.
Defining H2. Yes. But because H2 is involved in a variety of physiological processes, that's not being pursued as aggressively as direct antivirals that act directly on the virus.
One more question, then we should go out because otherwise you'll miss all the wine.
Yeah, so I'm I find your models very interesting. For somebody who hasn't seen this at all before, I find it morbidly satisfying. So I'm curious though, with the development of the mutations, the new variants, can you point to specific variables in the model that translates to?
Yes. Yeah, so people talk about the variants being more infectious. So that translates into this parameter beta. Some reports have said that the variants they generate higher viral loads. So the simplest way in our model would be that the viral production of infected cells would be the birth size and the production rate P. We've been looking at, again, data from the same group who did the NDA double data. They put online 7 patients who were infected with B117 and tried to compare them with other NDA data set. I didn't have that. And at least at the level of resolution of 7 from various samples of 7 from this 49 in there, we see no statistically significant difference that we can pick up. Sample size is pretty small. I happen to know the grapevine and now are up to 13 or 14 B117 patients. They've also concluded that they don't see any statistically significant difference in genetics in that sample. Although they had a preprint with the 7 saying that they thought they were different, but final analysis that they wanted. Hour-long conference call with them, told them there's a problem with their conclusion, and they finally agree. But we really need much larger data sets. We're looking now at data of the use of monoclonal antibodies. So Eli Lilly and Regeneron have monoclonal antibodies that are used therapeutically. The first Lilly drug was withdrawn from emergency use authorization because of various drug-resistant variants. And the one that pops up is something called E484K, and that's the one that South African variant and the Indian variant carry, and it's a spike protein, and it affects antibody binding, and it was selected by these antibodies. So we're trying to understand the kinetics of what's going on with viruses. But again, we only have 6 patients, data from 6 people who generated that mutation spontaneously, which is different from the ones that are being transmitted. So they're selective in the presence of antibodies. So that's an evolving story. We're only into it for about a week and a half so far.
Okay, well, thank you, Aaron.
Sure, thank you. Yeah, it's the easiest one.